everybody, this is Miss Segovia and I'm going to go over your notes for viruses. So let's go ahead and set this up Cornell style. Give yourself a little room on the side to do your questions and then a space at the bottom for your summary. So our title today is viruses. Go ahead and zoom in for you here. So what is a virus? So viruses are particles of nucleic acid, protein, and in some cases we have some lipids on there. And what's really interesting about viruses is that they can reproduce only by infecting living cells. They cannot reproduce on their own, and per our definition of what's life, this means that viruses technically are non-living. So a typical virus is composed of a core of either DNA, let's underline that so we remember DNA, or it can have RNA in there. Those guys are called retroviruses. And so which is surrounded by a protein coat or what we call a capsid. So earlier, hopefully if you haven't yet, you did the little viral infection. And so what we're talking about here is this little area up on top. So this is what we call our capsid. And so that's the little protein around the outside, and on the inside here I have colored red is our DNA or our RNA. Now not all viruses look like these cool little spaceships. Some of them are round like balls with little pokey things out. Some can be kind of look like squiggles. Ebola's kind of a cool one, looks tubular. Lots of variety in viruses. So when a virus infects, when we have a viral infection, there are two different cycles that it can go through. So one, we can have a lytic infection, or we can have a lysogenic infection. And that's kind of what the picture you guys have on the left is about. So let's go ahead and do this, and we'll do a little partition down here. So when we have a lytic infection, our virus enters the cell, makes copies of itself, and causes the cell to burst, which is also known as lyse, and release, L-E-A-S, more viruses. And so the lytic cycle is what we see on the opposite page on the top part here. And so the virus infects the cell. It'll actually become part of the cell's DNA, and then it makes the cell make more viral proteins and assemble the viruses, create a whole bunch, and then release. It's kind of like a good science fiction movie where, you know, Invasion of the Body Snatchers. They take over and are in control of the cell and make it do whatever it needs it to do. The other type of infection is a lysogenic infection. So in this one, The virus integrates, I'll zoom in on that for you a little bit. The virus integrates DNA into DNA of the host cell, and the viral genetic information replicates along with host DNA. So what's really weird about this one is it's the lower half that you see over here. And so the DNA becomes part of the cell, and then as the cell grows and divides, the viral DNA stays in there. At some point, the virus can exit the lysogenic cycle and go into the lytic cycle. And so cold sores are a great example of this. It'll lay low and just replicate, and then at some point it decides, you know what? Let's have a nasty cold sore show up on some poor person's face. And so it'll come out. A lot of viral infections go this way, and they're so successful because they can hide in there and stay in there for a very, very, very long time, or even for the remainder of your life. So there's several different types of viruses. Let's go ahead and just box this off here so it looks a little bit nicer. We can have what are called bacteriophages. So a bacteriophage is a virus that only infects bacteria. So if you're in the presence of a bacteriophage, don't worry too much, because if you're an E. coli, you should be shaken, but everybody else would be okay. We also have a prophage. So prophages, those are the ones where the viral DNA is embedded in hosts' DNA during lysogenic infection. So like I said, that's kind of like your the cold sores and herpes and stuff like that. They're prophages. And then we also have retrophages. And so these are viruses containing RNA, which will then produce DNA of their RNA upon infecting a cell. And so rather than carrying DNA up inside the capsid, they actually carry RNA. And so once that RNA gets in the system, it kind of gets reverse transcribed into DNA, and then that DNA will go ahead and that'll be used to create our new stuff. And actually, I just realized I made a mistake. Instead of retrophages, it should say retrovirus. I got so used to putting phage, V-I-R-U, virus. I'm really doing great today. Retrovirus. All right. So when it comes to looking at viruses and cells, we have a couple different characteristics that we like to compare. So for this one, we're going to do a little Phagen thing. So I'm going to pause here and come back in half a second and show you how to do that. Hey everybody, for this last little part, we're going to put more information that can fit on the page. So we're going to do it on a note card. So rather than you watch me write, I'm just going to zoom in and walk you through what's going to go on your card. So we're going to look at the difference between a virus and a cell, the characteristics of viruses and cells and how they compare to one another. So the first part is our structure. Viruses are composed of DNA or RNA, a core, and a capsid, whereas cells, especially eukaryotic cells, have cell membrane, cytoplasm, nucleus, and organelles. Let me make that T on cytoplasm a little more obvious there, cytoplasm. The reproduction of a virus is only within a host cell, whereas cells can reproduce independently. So viruses must rely upon a cell in order to make more viruses, where your cells can just reproduce and grow on their own, usually through the process of mitosis. The genetic code, viruses can have DNA or RNA, cells are just DNA. Growth. Viruses, once they're produced, do not grow any larger. You can't have a baby virus. It's just a virus. Whereas cells can start out smaller and grow. We can have development in multicellular organisms. You start from a single cell as an embryo and grow into what you are today. When it comes to energy, obtain and use energy, viruses don't need to. Once they take over the cell, they use the energy that's available there to reproduce, but that's it. Regular cells, though, we need outside energy, or we need to be an autotroph and produce our own energy. Respond to environment. Viruses do not. Doesn't matter if you're too hot or too cold. They will mostly be okay unless you get so hot that you denature the proteins, whereas cells are very dependent and do respond to the external environment. And finally, change over time. Both viruses and cells do change over time. The fancier word for that, of course, is evolution. And so we do see evolution in viruses and we see evolution in cells. So with this little note card, what you can do is fill it out and see how it hangs over just a little bit. You can go ahead and put a piece of tape on there, and then underneath that, if you want to include any additional information or look up some cool virus pictures to include in your notes, that would be helpful for you to study. But this way you've got that little table, and you also have space to do your summary, so on and so forth. So that concludes your viral notes. If you have any questions, please let me know. Thank you.